Morning, guys. How y'all doing? We're going to try to run through 16, 17. We get to the near the end of the race here. We're going to go through 19 in General Biology 1. These last four chapters, some of them we'll be jumping in lecture because we don't have time to go over all of them. But here's one that I wish we had time to look at. This is chapter 16. So I'm going to share my screen with y'all, and we're going to run through it. It's going to be a pretty quick run through, so don't stress out. It's not that long of a chapter. I'm going to move my picture right here so I can see. Make sure I'm looking at y'all when I'm talking. There we go. All right. So if you can see my screen, this is how populations evolve. And I only have like 7 questions, I think, on this short answer. It starts off with a real bang here talking about gonorrhea, a bacterial sexually transmitted STD, right, that has become resistant to antibiotics. So overuse of antibiotics result in the evolution of strands of gonorrhea that none of our antibiotics work for. And so that's a bad way to die, right, of gonorrhea that we can't treat. Okay, that would be a bad way to go. And this is a good reason to kind of go into a kickoff as to looking at population genetics is what this chapter is about. So the population then is a group of organisms of a single species occupying a particular area at the same time. Okay, population genetics then is the study of the diversity in terms of allele frequencies. Okay, and we're going to actually see, if you look in our book, on the first page of the chapter, page 280, we get one of my favorite terms, and that is, I already hit it earlier about the 2 types of evolution. Microevolution, which is changes in allele frequency of a population over time, versus macroevolution, which is that these small changes over time lead to the creations of a new species where we have a separation of a species, and that's macroevolution. We're going to talk about that in another chapter coming up. But in this one, we're going to be looking at microevolution, and that is changes in allele frequency over time in a population. So the various alleles at all the gene loci and an individual make up its gene pool. And gene pool of a population can be described in terms of the genotypic frequency, remember, versus phenotypic, what you express. We're going to be looking at allele frequencies. This field first got popular in the 1930s, right, by population geneticists. I actually took a class in this in grad school in population genetics. I found it quite interesting. And the example that Bader is going to use is one of my favorites that I mentioned already, the peppered moth of England. I told you about how they had evolved over the last century and a half or so. Well, now, probably 120 years we've been watching them, and we've seen them shift from a white phenotypic population, big D, little d, is the homozygote, the white colored moth, to, sorry about that, I hit the wrong button, to this darker moth, which is dominant, which is big D. Big D would be dark moth, and big D, little d heterozygote would be the dark moth also, okay? Now, we're going to play with some numbers here, so you got to do a little math if you're going to do population genetics, okay? So let's take a look at it. If the proportion of each allele within a population is a real frequency is the proportion of each allele within a population in a gene pool. Okay, so it can be expressed as both the dominant and recessive alleles added all together will equal 1. So percentage-wise, so 0.5 would be 50%, big D, 50% little d. Okay, so P and Q. P is usually, it can be either one allele and Q the other, and there's no rule that one of them's got to be the dominant recessive. But we usually, I think we'll see that there's a pattern there. Okay, so in microevolution involves a change in these allele frequencies within the population over time. So that would be microevolution. So take a look. So we're going to look at a pretty wild number here. Look at this. We're going to look at a population of 25 moths. Okay, and that means they have 2. Each moth has a gene from mom and dad, right, for that trait, for the color. So there'll be 50 alleles. Okay, and in this population, we're going to have 16 of the white, homozygous, recessive, little d's. Okay, so if that is true, check it out here. So we would have how many? We would have, count them up here. We would have, if I got it right, 16. We would have 32 of those alleles, right, of our 80. Okay, and then take a look. And then up here, we're going to have, in this population, we've got one big D, big D, and then 1, 2, 3, 4, 5, 6, 7, 8 heterozygotes. So if you add up those total alleles, you end up with 10 big D's and 40 little d's. Okay, now put that in our equation here. So 0.4, right? So 4 out of 40 out of 50 would be what? 80%, right? So the big D is 40 divided by 50 is 80, and 10 divided by 50 is 20%. So our Q is 0.8, and our P is 0.2, representing 20% and 80%. Now here's where the Hardy-Weinberger equation comes in. If you take these percentages and put them in this equation, P squared, take that P number, which would be this one right here, 20%, and square it, which is 0.2 times 0.2, that would lead to 0.04% of the population being homozygous dominant, or 4%. Q squared, which was our other number here, and I see a typo down here again, it's not 0.08, it's 0.8. So 0.8 times 0.8 would be 60.64. So 64% of the population would be heterozygote. And then our middle number right here, the 2PQ, represents the heterozygotes. So P times Q, 0.2 times 0.8, it'd be 0.16. 2 times 0.16 is 32. So look at the math here. So 32% of our people will be heterozygotes, 4% will be homozygous dominant, and 64% will be homozygous recessive, which is what we saw, or what we see in that group. Okay, is that not kind of crazy? It really is. Okay, so when you look at it together, then we got the genotype and phenotype. But this will give you the phenotype. By putting the numbers in the equation, we can get the percent of each phenotype we would see. Pretty crazy, or genotype we'd see. Okay, now here's where this is cool. Hardy-Weinberger equilibrium says that allele frequency in the population will remain constant, assuming these 5 characteristics are met. So evolution will not occur, the allele frequency will be the same, and microevolution will not occur if you reach Hardy-Weinberger equilibrium. And to achieve that, you must do 5 things. You must have no mutations, no gene flow, which we'll define that in a minute. That is organisms entering, leaving a population, immigration, migration. You must always have random mating, which we know never happens in nature, and no genetic drift. And we're going to talk about that in a minute. Genetic drift is random changes in allele frequency due to chance, not natural selection, but just random chance. Earthquake, storm, fire, whatever. And then no natural selection. Natural selection is when nature selects for a trait that gives them an advantage of surviving in the future. Okay, as we've been talking about Darwin's that. So no natural selection, no genetic drift. No, you have to have random mating. Okay, because non-random mating would lead to changes, right? But pure random mating, no gene flow, and no mutations. Okay, so 4 no's and with the word. And so these 5 things are met, then you would have no evolution, no microevolution occurring. Okay, so let's go back to our pepper moth, and here it is. So we got this population, right? There's 4% big D, 16% big C, big D, little d. Sorry, trying to touch it on my screen. And 16 is 32% of that, right? And then 32% heterozygote, 64% homozygous recessive. Okay, so Hardy-Weinberger equilibrium requires conditions are rarely met, if ever in nature. Deviation from Hardy-Weinberger equilibrium indicate that evolution has taken place. And by looking at the actual, when you see a change in allele frequency and we know microevolution is happening, then we can go back and find which of these 5 things happened. Did mutations occur? Did gene flow occur? Was there non-random mating? Was there genetic drift? Was there natural selection? So these are the 5 agents of evolutionary change, okay, which are the 5 things that must not happen in order to have Hardy-Weinberger equilibrium. So it's kind of a cool concept here. So let's take this is a pretty cool table. Look at this. So if you have random mating, that's Hardy-Weinberger equilibrium. The opposite would be what? Deviation would be non-random mating. When you have non-random mating, alleles do not assort randomly. Changes in genotype frequency occur, but that doesn't lead to necessarily evolution. Natural selection, however, certain alleles are selected for or against. Change in allele frequency over time will lead to evolution. Mutations and new types of alleles, change in allele frequencies, mutation occurs. No migration, but if you have immigration and migration in and out of the population, then individuals carrying alleles into or out of the population will change the allele frequency. Evolution will occur here. And in large populations, you get no genetic drift. But if you have small populations, we're going to look at 2 famous types of genetic drift, where you lose allele diversity, and some alleles may even disappear, and this can lead to evolution. So these 4 can, and over time, I was under the impression, I thought that non-random mating can, but maybe it doesn't, okay? All right, so let's go back to our pepper moth and take a look again. So here's our original numbers, right? 80% homozygous dominant, homozygous recessive, white, 20% dark alleles, with 4% big D, 32% were what? Heterozygote, and 64% was the little d, white moth. Okay, now here's what happens. We breed all of them to each other. We have random mating, but no selection, no migration, no mutation. Then we expect the allele frequency stays the same, but the frequencies change. But see, that's not changes in allele frequency. The frequency change doesn't change in random mating, but you do get variations, changes in the phenotypes. Okay, so no changes in allele frequency, no change in genotype, but we do have some phenotypic changes, right? Has it moved anything yet? Actually, no movement here yet. So nothing's happening here. Okay, yet. So nothing happened. Sorry, I thought we had some changes there, but we didn't. Okay, now that was with random mating. If we have non-random mating, because remember, random mating is one of the requirements for Hardy-Weinberger. Non-random mating, meaning selection occurs among individuals. That doesn't lead to new genes, but it does change the phenotypes. So no change in allele frequency. The genotype frequencies change. Evolution has not occurred, even though we've changed color. We haven't evolved any changes in allele frequency. Natural selection, where the white moths are eaten, and we come back and now we see 80% dark and 20% white moths, and now we put that together, and now this thing is flipped. So now white moths, little d are 4%, heterozygotes are 32%, and 64% are dark. Then we have seen a change, a mega change in the allele frequencies, right? So the allele frequencies are flipped. We've gone from 20% Ds to 80% Ds, and that is microevolution. So even though we can have changes in the phenotypes, the genotype combinations changes, we haven't changed the allele frequencies. And that is the definition of evolution. Okay, pretty cool. Pretty cool. Okay, so just going to go through these real quick. So some causes of microevolution. Obviously, mutations lead to new ones, and that's an easy one, right? They provide new alleles and new structures, new pigments, new whatever. Gene flow is a kind of a cool one, where we have organisms moving in and out of a population. Okay, and that's definitely going to change the gene flow, the allele frequency. I used to tell a bad story about, remember, the tongue curling is one gene, big C, and big C, little c can curl their tongue, little c can't. And so if you had a county that had 50% of each in the gene pool, and it's a bunch of people that could curl their tongue moved in the area, then the allele frequency of curling your tongue would go up, and that would be gene flow causing evolution. I used to tell a story about that. Look at that. Gene flow could be the pollen being blown over across a river. Okay, now, another cause of microevolution, genetic drift. And this is a little bit trickier here, when to holler. Changes in allele frequency of population due to chance rather than selection. So it doesn't necessarily lead to adaptation to the environment, but it occurs due to some disruption by some random sampling from the population. So what if you're a rabbit living near the river and the river floods and you drown? That had nothing to do with selection. It just, you picked a bad place to live, okay? And we have two examples of this one. There's a short answer question on your group. Bottleneck is when it's when you have something happens and the number of organisms die and it gets real low, and then the only ones that survive are the new gene pool. And we'll see an example of that. And then inbreeding, and then founder's effect. Let's take a look. Here's the one they like to show in genetic drift. You have 10% of the population is the white, and 80%, I guess, is dark. Fire, and you had a population of forest fire kills 5 light moths, and you had a total of, look, you got a total of what? 10. You have 10 moths. One of them is dark. Fire kills 5 of the light moths, and now you have the one left with 4 others. So the population went from 10 to 20% to the light on the dark moths. Okay, so that's genetic drift. Okay, bottleneck is a random event. A random event prevents a majority of individuals from entering the next generation. Okay, we have a good example of the bottleneck right here, I believe, right here. So here's a good example of bottleneck and founder effect. If you had 380 each marble represents 10 alleles. So if you have 38 alleles, 380 of these marbles, okay? And look at all that diversity of color. You have 53% blue, 26% green, 30% red, 8% yellow. And but then you're only going to have 9. The first 9 fall out. And now look what happens. These are all the genes we got left, which would be 90 alleles, 10 for each. You have now you've had 44% go to green. The blues are down to 45. Green goes way up, and red falls. So that would be bottleneck. Okay, and then founder's effect would be whoever founds the population. I always make a joke about this. Think of it like this: if we were going to populate Mars, and this is kind of social media when you may not have heard of these stars because I'm getting old in my old age. But Whitney Houston was one of the great singers of all time. I actually saw her in concert when early in her career. She had a beautiful voice, and she was one of the great singers of all times. And Elvis Presley, I did not get to see Elvis, but he was from my hometown. We share a hometown, so I've been a big fan of him my whole life. And he was one of the great singers of all times. So let's just say we could clone Elvis and who is dead, and Whitney Houston, who died a couple of years ago, clone them and get them in their prime, let's say at 28-year-old, and we send them to Mars to be Adam and Eve of Mars. What could you say about Mars singing ability, the Martian singing abilities? You would say that there would be some serious singing in Mars, right? Now, they both had drug problems, so there'd be a lot of drug dependency in there, and in Mars too. So that goes with the good with the bad, okay? And they were both good-looking people. I think most people would agree. So anyway, so whoever founds the population, your genes become the norm, okay? And a bottleneck, some event like the fire, can cut the numbers down and change the gene population too, and cause for evolution. Random mating, non-random mating doesn't happen in hardly in any species, any population we've ever studied, okay? They talked about on the island of this island, non-random mating results in increased frequency of color blindness, okay? That's pretty wild, okay? Natural selection, the one we done talked about, you have to have variation, inheritance, we've proven that. Differential adaptiveness, right? Some trait gives them a better chance of survival, and then those who have a better chance of survival breed more, and their genes become the norm over time, okay? And all those will lead to microevolution, okay? Natural selection, because of its polygenic inheritance trait, you can get a bell-shaped curve on that, and we see three types of this. We can see directional selection that would lead to a new species, possibly, right? Where you have a movement toward certain allele frequencies. Or we can see stabilizing selection where something works, like 5 fingers, 2 hands, and then it becomes the norm, so it gets a real compressed curve, we're going to say in just a second. And disruptive, and where 2 extremes of a characteristic are selected for, and it's very rare in nature, okay? And so what I'm saying is you would see a directional move, right? Or in this case, you could see that's directional here, stabilizing or disrupting. Okay, here's a classic example of stabilizing. This is a cool graph in a lot of the books. This is the percent of birds populations in this weight zone. And by the way, this right here, 2.7 to 3.2 kilograms is around 7.5 pounds. Look at that, 20%, 15, almost all the babies are born between what? This 1.8 and 4.1, right? And then look at the red line, that's the percent of infant mortality. So if you're born real premature, then your chance of dying are 70% on that one, 30% on that, and that means not making it to first birthday. But you get down here in this sweet spot, right, and it's 2 to 3% death rate. So nature has selected for a birth rate in humans of around 7.5 pounds, and that gives them the maximum ability to live to make it to the first birthday. That's pretty amazing. Of course, if you're a mutant weighing 30 pounds over here, that's going to kill mama and probably not healthy either. So it's not 30 pounds, that's a big kid over on that end. Directional selection would be a bacterium here. These two are killing everybody else, right? They're drug-resistant strands, right? Not drug resistance, and you expose them to a drug and it kills them. And then over time, this non-resistant group take over your body. So when you take antibiotics and don't take them to the very end, then any antibiotic-resistant groups in your body then take over your body, and that can lead to big problems later. Okay, and here's the classic one, this land snail. I believe it's called, I'm not sure what it is, the British land snail. I don't think it was British, but anyway, kind of an interesting group. You have 2 extreme colors. You have the brown stripe with yellow stripe here, and it loves to live in the ground, in the gravel. And then you have this one with the darker bands that are a little bit darker in the spiral here, and they live better in the limbs. Intermediates get eaten by the birds. So when they breed together, anybody that's got both of these mixed together get killed. So we see a disruptive selection to 2 extremes, just one of the rarest ones in nature. That's disruptional natural selection, where it pulls 2 extremes. Be like nobody would be average height. They'd have midgets and giants. That'd be the equivalent, which that doesn't occur in any human population, right? But in these snails, it does. Sexual selection, right? And this is a wild one here, that there is sexual selection in nature. With males, there is an increased ability to compete with other males for mates, and females choose a mate with the best fitness. Fitness is defined as ability to produce surviving offspring. This leads to some crazy stuff, including female choice, where females choose can be explained why male birds are more oriented, more beautiful, have more coloration because they're looking for the prettiest one that has the best health, or their plume is very beautiful. And then we see, there we go. So here the drow female at the bottom tends to choose flamboyant males as their mate with these beautiful colors. Okay. And then male competition, where we've all seen the chimpanzees fighting and hollering, and of course the deers screaming, they all hollering at each other. This red mule deer banging horns, fighting right for male competition, and the winner gets to do more than breeding. So the top of the food, the male dominance hierarchy gets to do more breeding in the population. And it just talks about genetic variability is important, so we maintain some variability. We don't want to become all the same. Possibly that's not good evolutionary wise. And natural selection causes imperfect adaptations that have to die out. So evolution is not perfect, right? It doesn't start from scratch. It just takes what we got and builds on these imperfect situations over time. And it talks about that, some more of that. Nothing much here. This talks about another one, how we can have subspecies of these are the garter snakes, and they're subspecies in in the southeastern United States. 4 different ones here that are subspecies. And then another wild one here is heterozygote recessive alleles have a purpose here where the heterozygote has a greater chance of survival. And the classic example of this heterozygote advantage is sickle cell anemia. If you have both sickle cell genes and you die, big time problems, right? But if you're a carrier, then, and of course those who have full 2 sickle cell genes, they have resistance to malaria. Malaria is caused by a protozoan parasite that lives and destroys red blood cells of homozygous normal individuals that have no sickle cell trait. Heterozygotes have an advantage, and this is a form of stabilizing selection. And if you look at this crazy map here, this is where malaria occurs. Yellow and the blue is where malaria occurs also, and the blue though is where sickle cell is red, and the blue is where sickle cell is very common. So where you see the overlap, the blue, both red and yellow are here. You have malaria and sickle cell trait. My daughter went to this part of Africa 2 years ago, and they went down and did some doing some wells for water and malaria treatment for kids. So malaria really prominent there, and those kids who have sickle cell trait, the protozoan can't live in their red blood cell and thereby can't kill them. So they have resistance to malaria. We also see, your book also mentions that we also see that in the people of European descent with cystic fibrosis. Okay, so there is a resistance to those who are carriers to that too. All right, and that's all I got here. Pretty short chapter here on 16. Just know your questions. Won't see a whole lot of that, and we'll have a few multiple choice out of the chapter. Y'all have a good day, guys. I'm going to stop sharing. We're going to call it a day on that one. Pretty short chapter. Y'all have a good one.